It's noon, and I think we should probably get started with the webinar. I'd like to welcome you all to the White Bark Pine Ecosystem Foundation's High Elevation 5-Needle White Pine webinar. And this is the first webinar of the academic year. For those of you not in academics, we tend to run this from fall clear to spring and then take a break for the field season. So I'm Diana Tomback. I'm professor of integrative biology with the University of Colorado, Denver, but I'm also a founding member of the White Bark Pine Ecosystem Foundation. And I'm now serving on the board as policy and outreach coordinator. So this webinar series on white bark pine and other high elevation 5-needled white pines is a project of the White Bark Pine Ecosystem Foundation. I think I'll refer to it as the WPEF for short. And I'd like to acknowledge Karen Nelson, professor at the University of Montana, for initiating this great educational and outreach activity. The first year of this webinar series was last year, and it was very successful. So a few words about the White Bark Pine Ecosystem Foundation. We are engaged in a number of activities related to science advocacy for white bark pine and now other high elevation 5-needled white pines. We promote knowledge concerning white bark pine and these other pines and their ecosystems. We will try to describe one of the projects that we're involved in as part of the introduction to each webinar, so you get a sense of the work that we're doing. So today I would briefly like to mention that we are science leads in a partnership with American Forests in devising and assembling the National White Bark Pine Restoration Plan. And our partners in this effort include the U.S. Forest Service, the National Park Service, Bureau of Land Management, U.S. Fish and Wildlife Service, and several tribal jurisdictions. As many of you may know, white bark pine has been proposed for listing as a threatened species under the Endangered Species Act. And we believe that a decision will be made about this listing within the next few months. Also, I will mention as partnerships, several tribal jurisdictions, and we are very pleased to be working across various tribes in the West. We anticipate having a draft of this very ambitious restoration plan early in 2023. Now, a word from your sponsor here. For those of you who are not members of the White Bark Pine Ecosystem Foundation, joining is very easy on our website, and it has a lot of benefits. We have a newsletter, this webinar series, a future webinar series, the Nutcracker Notes Journal, which is exclusive in the first few years to the membership, opportunities to present research at our meetings, but more importantly, you going on record as a member provides grassroots supports for our efforts and our outreach. We can point to a supportive membership. And I'll also mention that student membership remains a very low $25 a year. There's not that much you can get these days for $25. So there's lots of reasons to join, and I hope you'll join today to support white bark pine and the other high elevation white pines. At this point, I am really pleased to turn the webinar over to our graduate student organizers, Vlad, Laurel, and Theo, to introduce our speaker. Thanks a lot.
Thanks, Diana. Hi, everyone. Thanks again for coming. I'm Laurel Sindewall, the PhD candidate in Diana's Forest Ecology Lab at CU Denver. As Diana mentioned, this is the first webinar installment of 2022. If you missed the webinar last year, you can find recordings of the talks on our YouTube channel. The link for that and for our website is in the chat. There we go. Done. Today's speaker is Jeremy Johnson, who is an assistant professor in the Department of Forestry at Michigan State University, and an affiliate researcher at USDA Darina Genetic Research Center. Dr. Johnson is a forest geneticist with broad research interests geared towards understanding the spatial and temporal patterns of how forests respond to multiple threats associated with global ecological change. He does this by specializing in the use of geographic, landscape ecological, and genetic tools and theory, with the goal of ensuring the long-term health of forests using conservation interventions and management. Dr. Johnson completed his PhD at Texas A&M University, after which he was a postdoc in the School of Forestry at Northern Arizona University and the USDA Durina Genetics Resource Center. Today, he is here to talk about the use of hyperspectral imaging combined with a custom motion control system and machine learning to identify and track the progression of white pine blister rust in white pines, with an aim to identify rust-resistant seed sources. If you have questions following Jeremy's talk, we encourage you to virtually raise your hand so that we can enable you to unmute yourself and start a dialogue. If you are unable to do so, you may put your questions in the chat and I will read them. With that, I'll turn it over to Jeremy.
All right, thanks very much. I'm going to go ahead and share my screen really quick here. All right, hopefully everybody can see that. So first, I'm very happy to be the speaker to kick off the seminar series this year. And what I want to do with this talk is sort of outline some of the different research projects that I've been working on with a lot of different collaborators over the past several years. And I'm going to focus on some of the work that I've done to identify genetic resistance to white pine blister rust in 5-needle pines. And also thinking about how we can improve the process of phenotypic evaluation. So how do we actually speed up and improve the temporal and spatial characterization of these white pines in order to better combat the interactions of climate change and this non-native disease, white pine blister rust. So we're all here for the same reason, right? We love white pines, high elevation white pines in particular. And so when you look at this map, you've probably all seen a map like this or something similar. This is the geographic distribution of our favorite pine group in section strobus, the white pines. In North America, there are 9 of these species, and they grow in different environments and conditions. And that, of course, ranges from the high alpine elevation, white bark pine being the classic example here, and all the way down to these lower elevation eastern white pine forests that are often dominate the xeric northern pine forests. So where I am here at Michigan State, we have a whole lot more white pines, eastern white pines. And they often are economically important. Eastern white pine is an example of that, sugar pine, western white pine, but they're also super important ecologically. When we think about white bark pine and the role that it has in ecosystem services and functions, it's very, very important. And so we can all think of reasons to care about these types of forests. And it's with this thought in mind that I want to talk about forest health and how we can better improve our integrated management solutions in a forest health context. So the Forest Service has estimated that 33 million hectares of forest land, this is almost 7% of all the forested land in the US, is at risk of losing upwards of 25% of tree vegetation in the next several years. And this is associated with non-native pests and pathogens being introduced, some native species as well. So what is it that there we go. One of those diseases is white pine blister rust, right? And this is caused by the non-native invasive fungal pathogen, Cronartium ribicola. It was introduced into British Columbia, around British Columbia in the early 1900s, and it spread very rapidly from that point on, encompassing most of the range of the native white pines in Western North America. It was more recently discovered in the Sacramento Mountains, in New Mexico in the Lincoln National Forest, where southwestern white pine is at its southern periphery. And southwestern white pine, Pinus strobiformis, is now infected with this disease. And what's interesting about this is that the range of southwestern white pine has a core range that runs through the Sierra Madre Occidental in Mexico, I should say, and then it has this disjunct periphery population that is in Arizona and New Mexico and some of Colorado as well. However, the disease has not yet been found in the core of the species range in Mexico, which leads us to be a little bit proactive with how we can deal with an emerging threat in one of these white pine species. Despite the fact that it hasn't been found in Mexico at this point, recently, as of last year, there was a rusted ribes that was discovered near Wilshire Peak in California, suggesting that this pathogen is continuing to move further south, and that it's probably only a matter of time until the disease does reach the core of the species range. So how do we deal with this sort of emerging disease, and how can we go about identifying genetic resistance to it? So before I jump into the phenotyping and the remote sensing component, I do want to talk a little bit about the biology of the pathogen and then the different types of genetic resistance that exist to deal with this disease, and also the role of climate change. So in general, the way that the disease progresses, Cronartium ribicola is a macrocyclic pathogenic fungus. It has 5 different spore stages. It requires an alternate host. It's got a very complex life cycle. And in general, what we see is that the basidiospores will infect the needles of the white pine. And when this happens, it leads to this sort of characteristic needle lesion formation on the needles, the spotting that is so typical of white pine blister rust. The fungus will then move through the cellular tissue of the needles into the stems and the branches and into the bole of the tree, eventually, where it will create these sort of incipient cankers. And as this occurs, the fungus will then turn into a pycnium, and it will exude these pycnial drops that have a high sugar content, which will attract insects. And this is the stage within the fungus where you have the development of genetic diversity within the fungus as the insects fly back and forth, introducing novel genetic characteristics to the fungus. Eventually, those incipient cankers will continue to expand vertically and horizontally, essentially girdling the tree, disrupting phloem transport. This is the mechanism by which the tree will eventually die, or it will kill branches off. But the acial spores then developed and will erupt from these blisters. This is where you get that name, white pine blister rust. It's the characteristic stage in the blister rust. And those spores are pretty durable. They can disperse quite long distances in the right conditions. And then the acial spores will infect the alternate host, which is typically a ribes species. And then the rust will amplify as uridial spores on the ribes in the area, infecting more and more ribes until conditions begin to cool off and moisture levels increase. And then they will develop these teleal columns, and then the teleal spores will germinate and reinfect the pines. So that's complicated. It's any wonder that this disease continues to spread, but it does. And in addition to the white pine blister rust itself, we have to think about the role of climate change on these white pine species, these high elevation white pine species in particular. Joan Dudney, in a paper last year, was looking at some different time periods in the Sequoia Kings Canyon area and tracked the sort of climate optimum of blister rust in this environment, and showed that between 1996 and 2016, that the climate optimum of the disease has now moved into higher elevations, which should concern us all, because now we're starting to get more into the range of species like white bark pine and foxtail pine, and there are some serious implications for these species and their ecological niches. So we've got the disease, we've got climate change. When we look at projections for the future, we can see that across most of the ranges of all the white pines, we're going to see warmer environments, and how that interacts with the genetics of resistance is of concern. One of my colleagues, Andrew Shirk, and others in our group developed this ensemble species distribution model for southwestern white pine. And what they were able to show based on these projections is that by 2080, we're going to see significant range contraction in the species through the core of the species range. Also projecting some potential for expansion into Colorado, parts of Utah, as long as we take into consideration the role of dispersal and the capacity of the species to actually track their climatic envelope. And one of the questions that we've had is, how will different forms of genetic resistance and the stability of that resistance hold up in a warmer environment? So Richard Snesko and others at Dorina and myself, we sort of did a quick experiment this last year to simulate the role of climate change on several species of white pine. We looked at white bark and southwestern, western, and sugar pine as well. And in the cases that we had identified homozygous dominant resistance to white pine blister rust, we could see if increasing temperatures would reduce the stability of that type of resistance, basically allowing the disease to overcome major gene resistance. And superficially, what we found so far in our heated treatment was that the big R homozygous dominant western white pine genotypes actually had significantly developed cankers in the heated treatments, which again suggests that the role of climate change interacting with genetic resistance is going to be of concern into the future. So we've got these two big problems. We've got a non-native invasive disease. We've got climate change. What are we going to do? Because really our goal is to get trees onto the landscape that have adaptive traits, that have resistance to this disease. And how do we go from identifying types of resistance to these integrative management solutions? And so I want to walk through some of those steps and talk a lot about the remote sensing aspect of it, but I do want to highlight the different types of genetic resistance that occur in these white pines, and then how we can sort of combine these things to better manage the forests for the future. So a lot of these approaches involve a lot of people, common gardens, they take a long time. Remote sensing approaches can allow us to improve phenotyping. We can do experimental and empirical work to identify different relationships with environmental components, and then genomics to eventually identify loci associated with resistance. So for the rest of this talk, I'm going to talk about the types of genetic resistance that exist that we've found so far, with southwestern white pine primarily being the focal species here, but we'll expand onto some others. Thinking about how we can identify the presence of infection in white pine blister rust pre-visually using hyperspectral remote sensing. Can we use this approach to also better classify trees automatically into categories of health from a phenotyping perspective? And then thinking about how this technology scales from greenhouse experiments to operational plantings and beyond. So when we talk about genetic resistance in these white pines, we're usually talking about two different types of resistance potentially. And the most common that we hear about the most is major gene resistance. This was the type of resistance that was identified first. And in general, what we're looking at is the development of stem symptoms and canker formation on trees. And when we're using quantitative genetics approaches and we're partitioning variants across these different types of trials, what we're really looking at are how offspring, usually these are half-sibling, open-pollinated progeny trials. And when we plant a bunch of these seedlings from these seed families, we can look at the segregation patterns in phenotypes. So from a major gene resistance standpoint, the expectation is if it's open-pollinated and they're half-siblings, that half of the progeny should have stem symptom-free phenotypes associated with major gene resistance. And so that would look something like this. And that's relative, of course, to susceptible families where every single one of them will develop cankers and eventually die. And we can test these ratios using things like chi-squared tests or exact binomial tests to look at the probability that the expectation for a 1-to-1 ratio is met. These are basically Punnett squares, if you remember those from biology. So the second type of resistance that is common is quantitative disease resistance. And this is a much more durable form of resistance and is associated with, in general, with 10 to hundreds to thousands of genes interacting in an additive fashion to increase survival over time. And so we can still think about this using the major gene example to start out with, but instead of thinking about stem symptoms, this is just percent survival. And the same would be true for major gene resistance. You would have a 1-to-1 alive to dead relationship with MGR because half of them didn't get stem symptoms. But quantitative disease resistance would still have some families that have reasonably high percent survival, even in the presence of the disease, right? And it might range over quite a few percentages of survival. And in some cases, it could even be higher than major gene resistance if you have very high quantitative forms of resistance. And this is all, again, in relationship to the susceptible families that will develop stem symptoms, will die very rapidly and have 100% mortality fairly quickly. And the phenotypes or the traits that are associated with quantitative disease resistance oftentimes can look like bark reactions where you have necrotic lesions around cankers, which arrest the spread of the fungus on the tree. And we know that this is true when we look at an example here. This is a paper that Richard Snesko and I put out this last year, looking at a subset of southwestern white pine in New Mexico. We had around 40 different families from 3 different forests. And when we look at percent survival and the mean severity of the disease over quite a long time, this is about 8 years worth of data, we see that there's a very large range in terms of percent survival. And in some cases, for example, the Lincoln National Forest in green here, there's a strong selection pressure for resistance. This is the stand where white pine blister rust was discovered in southwestern white pine. But even in the other forest stands, the Cibola and the Santa Fe, we still have around 10% of the seedling families that exhibit pretty high levels of survival over a long period of time. You can see those a little bit right here. And when we look at the traits that they're associated with, it's usually fewer stem symptoms, a higher percentage of bark reactions overall, equates with improved survival. And across the entire range of southwestern white pine, this is some data that is still in the operational trials. We still see similar patterns, right? There's a nearly continuous range of resistance that occurs across the geographic range. Even in the case, the Sawyer 17 example right here is mostly in Mexico, where we don't have the blister rust, so these are naive samples, and just by randomly sampling the maternal trees across the range, we still see the presence of putative MGR in the dotted lines, and then a range of quantitative resistance, which is exciting. So the way that is done from an operational standpoint, these seedlings, the seeds are collected from the maternal trees. They are brought to the Dorena Genetic Resource Center in Cottage Grove, Oregon. They are grown for 1 year in a greenhouse if it's going to be a major gene resistance trial, or for 2 years if they become operational. And at that point, they're put into these frames outside, where they're grown for another year before they're inoculated. Once they're ready to be inoculated with blister rust, we pick those things up with forklifts, put them on trucks, and we drive them over to the inoculation chamber, where these metal frames are placed over the top of them. The rust crew will go out and collect infected ribes, and then the ribes leaves are placed teleal column side down over the tops of the pines, and we can monitor things like the spore load and the germination of the fungus using some sticky slides and some agar underneath the frames. And then we tent them, and we can modify the climatic conditions so that the temperature and humidity is at the right level so that infection will occur. And we have, as Richard will point out, and others that work at Dorina, have a very, very high success rate at infecting white pines with blister rust. But once that's done, the real work begins, because then you need to monitor the phenotypes that are associated with identifying resistance. So that would be counting all of the spots on the needles and counting the number of cankers and the severity of the infection, how much of the bowl is covered by a canker, what is the height and the increment growth rates, and all of these different phenotypes. And counting each one of these trees in a frame at Dorina can be very time consuming. And ultimately, it's very expensive. And we have issues of assessor bias. How are ratings determined when we have 4, 5, or 6 different people rating a trial across different reps? So this really brought up the question about how can we improve this process of identifying genetic resistance, improving phenotyping on an operational scale to improve the identification of elite trees that we can use in restoration and reforestation. And the answer to this question for me has been remote sensing, and using this new technology to sort of automate and operationalize the phenotyping. So what are the remote sensing approaches? When we think about remote sensing, using something like a camera, something that is remote from the object we are assessing, a simple RGB camera that has red, green, blue spectra on there would be the first way to do that, something like your iPhone, right? Now, there are few bands that would be used in this type of remote sensing, and so possibly using something like a multispectral approach that has a wider range of reflectance being measured might be a better approach. Sometimes the bins in which the spectra is collected can be wider, so you have some improved resolution spatially and temporally and spectrally to do this. And then lastly, hyperspectral. And hyperspectral remote sensing really contains a much larger range of reflectance and spectral wavelengths that we can look at. So to state it simply, hyperspectral just means that it's looking at more colors. And ultimately, it's detecting these colors beyond the range of human vision. So here's an example of what we might try to do. You can see the wavelengths from 400 to 1,000 nanometers in the electromagnetic spectrum, where visible light is between 400 and 700 nanometers, and you have the near infrared. And the red line right here is infected, and the green line is non-infected, right? It hasn't been infected with the disease. And you can tell that there is separation around the 600, 700 nanometer wavelengths. And what we can tell also in the near infrared is that there's good separation. And when you look at the images right here, you can tell that one is green and one is dying. Now, the important part here is that it doesn't actually take hyperspectral remote sensing to look at these two images and say that they're different, right? We can do that visually. But that's not always true. So in this example down here, we have 2 white pine that do look reasonably healthy visually, and we still have good separation based on the reflectance profile in the hyperspectral imaging. So that's sort of the background on this. And there are a lot of different aspects in biology of these plants that would affect the spectral signature, right? The biochemical and the biophysical properties of the plant. So different pigments and the thickness of the leaves, the chlorophyll and mesophyll, all of those have a bearing on our ability to separate plants that are infected versus not infected. So this is a complex process, and there's a lot of steps involved, and each one is important. The general approach is to start with target extraction. When we use a hyperspectral remote sensing platform, we are imaging the whole area, so it might contain background or a bench if you're in a greenhouse, the plant, as well as anything else that's around it. We need to extract the pixels that are just associated with the individual trees. We also need to be able to separate trees that might have overlapping canopies or might be very close together. So that first step of target extraction is challenging to automate. Next, we need to identify the particular spectra and the wavelengths that are associated with blister rust identification, from that full spectra of images. And then they need to be labeled and ground truthed. Once that's done, we need to go through identifying feature extraction. So can we actually identify particular bands or vegetation indices or ratios of bands that best allow us to identify infection? And then from there, we want to go into classification so that we can go beyond just identifying the disease, but also grouping the trees into different health categories that would aid in further looking at patterns of infection and survival and mortality through time. So there's a lot of different steps that are involved. So what I want to talk about are two different projects that have been ongoing using these approaches to harness automated phenotyping using hyperspectral remote sensing on the identification of white pine blister rust. So first, can we identify infected seedlings, right? What is the susceptibility of the plants? Next, how do we classify seedlings into appropriate health categories and what hyperspectral features do this the best? Third, what types of models in terms of the selection and timing of the models are most appropriate, because plants vary spatially through time. We need to be able to use the right machine learning approaches to automatically classify these individuals into the health categories. And then thinking about how this scales up from simple greenhouse experiments to operationalizing it in the field. So just to touch on what the sensor will be, we're using a head wall nano VNIR hyperspectral camera. There's 271 bands. The range is 400 to 1000 nanometers. And it's a push broom sensor. That means that instead of like your iPhone, again, you actually have to be moving the camera across the scene to capture the image. Okay, so the first part is looking at using hyperspectral imagery to detect an invasive fungal pathogen and symptom severity. And this is work that Maria Hagsma, as part of her dissertation, she was really instrumental in making this project so successful at Oregon State. So what we did was we took 175 seedlings from 10 different families that were spread across the geographic range of southwestern white pine, and they had different known levels of susceptibility to white pine blister rust. So we had 2 or 3 that had putative major gene resistance. We had some that we thought had pretty high levels of quantitative resistance. And then we had a range of others from susceptible to those that we didn't know about in Mexico. We separated these and split them in half so that half the seedlings were inoculated with Cronartium ribicola, and the other half was left as uninoculated controls. And then we put them into a climate-controlled greenhouse at Oregon State and monitored the progression of the disease. And we did that using this hyperspectral remote sensing platform that we engineered. We called this the HyperRail. What you can see here is that we put a piece of aluminum rail across the top of the bench. We 3D printed this housing for the camera. We have a step motor and an Arduino board in there that we can control the rate of speed that the camera moves across the scene based on the amount of ambient light coming in, so that we have equal exposure across the scene. And we took 16 different dates to acquire the hyperspectral images, and then we have corresponding visual assessments that were based on Dorina's standard MGR trial assessments, where we would come in and look at the number of spots, cankers, and we would classify the seedlings into these health categories: vigorous, moderately vigorous, sickly, and then dead from rust. But the basic approach, there's a little cartoon right there. The camera moves across the scene, and then as the plants change in terms of health status, we should be able to look at that. We should be able to tell the difference between individuals that have been inoculated and left uninoculated as controls. So here is a sort of temporal schematic of the progression of the experiment. We've got the 10 families plus all of them combined here. The arrows represent the 16 dates that the images were taken. And then you can see the proportion of families that fall into the different categories based on visual assessments. So in terms of major gene resistance, we can see those right here. They stay fairly healthy most of the time. We have some mortality. Same with this family, family D. And then we have susceptible families where they reach mortality fairly quickly and exhaustively. So what we found using the sensor is that hyperspectral imaging was able to identify 83% of the infected seedlings early on. That's important to note, in particular, that we were able to do that when we were only identifying around 20% of them as being infected visually through the traditional phenotyping approach. And so that's important. We are able to identify pre-visual infection using hyperspectral imaging. So we then used a support vector machine algorithm with a linear kernel to do a machine learning approach to allow us to classify these seedlings into these health categories. And we used the visual assessments as sort of the true status of their health. So using this machine learning approach, what we see is that 0 are the controls, they're uninfected, and then we have our health categories there. And we do a reasonable job at looking at the healthy category, the vigorous category. And of course, we do a great job of picking out those that are dead. The problem emerges when we look at the sort of middle categories, moderately vigorous to sickly. And what is actually happening is that we are incorrectly classifying the sickly and then the vigorous, right? Some of these are actually, that are classified as 2, are actually healthy or actually sicker than what the machine learning is predicting. And so that makes sense because there's quite a range in color variation. And when you're looking at these things visually, it's a little bit subjective how they're being placed into these categories from a human perspective. So we decided to combine those two classes together, a composite 2 and 3 health status, and we improve our probability of detection quite a bit at that point. And overall, we have almost 80% classification accuracy using this machine learning approach. So from this point, we're trying to figure out which portion of the electromagnetic spectrum using the hyperspectral camera best captures infection with blister rust and allows us to assign health as a category. And so to do this, we used a sort of 2-prong approach to rank some 88 different features. And when I say features, we're talking about, again, band combinations or simple ratios and normal vegetation indices that you might expect, like the normalized difference vegetation index. So first we used t-tests to compare the different indices and look at their classification accuracy. And we ranked them based on their p-value. And that gets us to this point where we're seeing the photochemical reflectance index or some derivation of the PRI, so the normalized photochemical reflectance index, or that which is associated with 570 nanometers. But even so, when we're using p-values, you can see that there are similar p-values on some of these indices. And so to to further separate this classification, we combined the p-value approach with the Bhattacharya distance, which is really a measure of closeness between two different distributions. And we use this as the second criteria to then further separate these indices. And ultimately what we found is that the photochemical reflectance index did the best job of pulling out and identifying blister rust and then aiding in classification of health category. And the PRI is associated with light use efficiency, photosynthesis, water content. So this makes sense when a plant is impacted by blister rust. The other point to make is that when we use the full spectra of the camera to identify infection or to separate vigor classes, we get 87% for identification and 80% for vigor class separation. And if we just use the normalized photochemical reflectance index, we see 83% and 76%, which is just a slight decrease in accuracy. But it's not so much that it doesn't make sense to sort of develop a more parsimonious platform that just uses the bands associated with the photochemical reflectance index, because then that camera can be deployed more cheaply and effectively than the expensive hyperspectral cameras. Okay, so I was mentioning that we used a support vector machine with a linear kernel as a machine learning approach for classifying these images. And the question is, why was a support vector machine chosen as the best machine learning approach? In a lot of ways, machine learning has become very popular, especially with highly dimensional data. But oftentimes what we see is that practitioners will neglect the practical aspects of selecting a machine learning algorithm. And when we think about plants, they're time varying in terms of how the traits emerge. And we need to think about what the most optimal machine learning model is for classifying these plants into these health categories and phenotyping. And so what we chose to do was use a k-fold cross-validation approach to test 22 different machine learning models. And these are all the models that are in the MATLAB machine learning toolbox. And so and using the default parameters in there. So we have models like random forests, k-nearest neighbors, support vector machines, trees, and they have different hyperparameters like the quadratic or linear kernels. So we can test all of those using a couple of different approaches. Now, k-fold cross-validation is a way that we can go and identify which model is the best, either using a predicted accuracy. And the way that k-fold cross-validation works is that it basically takes some number, k, of equal-sized subsamples of the data, and then uses that to train the model, and then you hold one sample out, and you predict it. And this is repeated however many times k is. So in general, what we've done is use 10, and this is often used as a heuristic in cross-validation this way. So what we found to start off with is when we use static data, that is, we basically lump all of the hyperspectral data into one distribution. So even though we have 16 different dates of collection, if we put it into a single distribution and we train the models on that data and then test it, we can develop this predicted accuracy. And what we find is that a support vector machine with a linear kernel with static data is the best model there. However, when we switch this over to dynamic data, that is, we use data from a single date to predict another date into the future, the predicted accuracy is not a very good measure of success in choosing these models. So the alternative approach was to use the area under the receivership curve and AUC. So this is another metric of model fitness, and in some cases it's been suggested to be a better measure. But AUC is essentially calculating the area of false positive rates versus the true positive rates as classification varies. So we can look at a classifier with high certainty. For example, right here, you can see the predicted values. We have high confidence in our prediction that a seedling is infected and the true value is infected, right? And over time. And when we do that, AUC is a better measure than predicted accuracy. And this is true for the dynamic data in particular. When we have low certainty, the predicted accuracy is higher than the AUC data. So when we look at our own data and we look at both the static and the dynamic training data, there's actually no significant difference between using predicted accuracy or the AUC as a measure of choosing a model. But with the dynamic data, there is in fact a significant statistical effect in that AUC is a better measure of choosing a model. Okay, so when we think about accuracy across time using predicted accuracy or the AUC measure for selecting these models, what we have here is we have different classifiers. We had 22 that we were interested in, and we also have these 16 test dates. So we have, and you can think about it, it's a k-fold approach. You've got 16 test dates and then 15 as a k-1. So we hold out one of those dates to predict it on. And for predicted accuracy, we see that we're above 75% in terms of classification accuracy using that approach. But AUC, again, has a slightly higher classification accuracy. And when we compare that to our support vector machine with the linear kernel, we see that it has the best results in terms of classification and identification. The one other approach to doing this is to use a heterogeneous ensemble approach. And there are different ways to do this with bootstrapping, but we used the approach where we chose the top ranked models and added them into a heterogeneous ensemble and then tested them using the predicted accuracy and the AUC. And what we see here is that in general, we have a higher classification accuracy using these heterogeneous ensemble models than we do using just the PA or the AUC on their own. And looking at the significance of that, the support vector machine still had the highest classification accuracy, but there was no significant difference between that support vector machine and the two ensemble models. But the ensemble models are better than single classifiers from AUC and predicted accuracy in general. And AUC is better than predicted accuracy when you're looking at dynamic data. So that was the way that we came about using the support vector machine with the linear kernel as our model in the original approach. So what we found was that early detection is possible and that the photochemical reflectance index is the best vegetation index in our study to identify the presence of rust, and that classification accuracy depends on susceptibility. So because of the range of variation in coloration associated with these different families of trees, we have some problems in the middle ranges, but we can do a pretty good job, around 80%, of accurately classifying these seedlings into health categories with machine learning. AUC is a more successful approach at identifying models than predicted accuracy, and the ensemble models are better than single classifiers. In our case, we chose the support vector machine from all those different models, but this is generally going to be case specific. So depending on the system and depending on the plants that you're using this approach on, you'll have to go through this process of choosing the right machine learning model. So we started out with this in a greenhouse in a very controlled environment, and the question had always been, how do we scale this up and do this over a larger area? And the Drina Genetic Resource Center was an ideal location to do this. They have many operational trials ongoing at various stages, and we could try to employ this approach for improving the inspections. And so what we did was we moved the hyperrail out of the greenhouse. We engineered what we call, it's a 12-meter hyperrail, steel extruded aluminum rail. And we still have step motors on here to control the speed of the camera, but it's scalable. And we also have wheels on the bottom so they can move across different environments. I have a video right here. You can see how it runs at Drina. And this is Maria in the image. And so this, I think, is either white bark pine or limber pine. But we have 3 replicates, and this hyperrail can scan. We were able to scan about 4 replicates on a pass, and then the whole rail will move down and scan the next set of boxes. So you're doing 4 boxes at a time, and we can usually scan an entire trial in about half an hour. And that's in contrast to the amount of time that it takes when technicians are out there, which can take up to a month. It's about 2 hours per box, an hour and a half to 2 hours per box. And we don't capture all the same types of phenotypes that the technicians are able to capture, but we can do this at a much higher rate through time. So we can track with a little bit more nuance the change in coloration and the change in health as the plants are impacted by the disease. And of course, this is not without challenges, because depending on the species that we scan, when we do white bark pine as an example, it's pretty easy to look at the separation of individual trees in these boxes. But once they grow larger and you have significant canopy overlap, it tends to be much more complicated in terms of how we do the image extraction and separate pixels out from one tree or another. So the goal of all of this is still really to get these resistant trees with adaptive traits associated with climate change onto the landscape. And so we really need to not throw away our ideas of conventional breeding and quantitative genetics. Those are very, very important. The most important aspect of all of this is good phenotyping, really. If you want to do genome-wide association studies and genomic selection and all of the new genomic approaches, you need to have very, very, very good phenotypes to go along with that. So employing high throughput genomics with high throughput phenotyping within a common garden approach, using machine learning to dissect the genetic architecture to look at quantitative disease resistance so that we can do QTL mapping and GWAS and eventually identify candidate markers for genomic selection. We really need to combine these types of approaches. And there are a lot more out there. We can use LiDAR to look at the structural components, infrared spectroscopy, chemical fingerprinting. There's so many different approaches that we can combine with the genomics and the variance partitioning to answer these questions. But ultimately, it's such a small part, right? Because what we really want is to get these trees on the landscape. So it's important that we test for resistance. But after we've done all that, we need to go out and protect those mother trees, the maternal trees. We need to collect and graft scion and put those into clone banks. We need to have durability and progeny durability trials and see how they actually hold up on the landscape. And so we need to continually monitor for white pine blister rust and other adaptive traits so that ultimately we're planting seeds that have resistance and adaptive traits, if possible, for real landscape level impact. Okay, it was a lot. And you know, it's sort of a complex topic when we talk about machine learning and hyperspectral remote sensing. And of course, all of this involved a great number of people. And I would like to thank Kristen Waring in particular and Richard Snesko, importantly. Chris Still, Betsy Goodrich, and Gerald Page were all involved in the STDP that helped to fund the Hyperrail project. And then Maria Hagsma did so much of this work as part of her dissertation. So I'd like to open it up for questions.
Awesome. So feel free to put questions in the chat and I can read them, or you can also raise your hand and Vlad should be able to enable you to ask the question directly.
Go ahead, Kara.
Hi, great presentation. Thanks so much. And thanks to all the organizers of today's webinar. Just to note that the chat is not enabled. So the only way participants can ask a question is by raising their hand.
I think Vlad fixed that, but thank you. All right, let's see. I see Enzo. I'm gonna unmute him.
Hello, Jeremy. Can you hear me?
I can.
Thanks so much for your talk. It's such a fantastic presentation, for real. I really like that you somehow just find a way to just present this in a really editable chunk and how to systematize early detection, and that's quite a challenge. I guess my question has to do with one of your earlier commentaries on how to deal with scale, because most of what you presented is like micro scale and how to detect like the different spectral forms of different levels of infections as well. And I'm interested in what are your thoughts of how to translate this micro scale early detection into a higher scale, and how could this be applied to the different levels of topographies? Like we have really accidented topographies like around Missoula or in the Western Rockies, pretty different to like Colorado or some other sides of the country.
Right.
And
Yeah.
how is like fit into the national restoration conservation plan?
Yeah, that's a good point. So it's always been the question, right? When I submitted the proposal to the USDA for this STDP grant, the idea that we were putting these cameras on drones had been problematic. They can be hard to fly. Sometimes they fall out of the sky. That happened to this specific camera. Actually, it cost like $15,000 to fix once it fell out of the sky. And so, before we started putting things onto drones, the question was, does it even work? So we started out in the greenhouse, and then it made sense to go to Dorina because the trees were small, right? This hyperrail right here is like 2 meters off the ground or so. And that's about as high as we can actually get it stably. So you're only able to use this with these smaller trees. And even if you look in the background up here, we've got trials that have trees that are pretty large, right? And you can't actually put this hyperrail over that very effectively. And so you do have to scale that up to something like a drone. It works very, very well in these situations where it's in a controlled environment. But the idea that we can take it out into the field is something that has to be explored more fully. Now, we were able to develop, and I should have mentioned it, this is what we call the mini rail, the mini hyperrail. This is the whitebark pine restoration site at Crater Lake outside of Rim Village. And we were able to take this rail there because, again, the trees are still reasonably small. You're not going to be able to fly a drone at the visitor center at Crater Lake, not very easily. You're gonna have to go through a lot of work to get a drone up there. So it was one way that we got there and validated in a more realistic setting, more natural setting. And then we've also taken it out to the Tyrell Seed Orchard in Oregon to see how it worked in those types of environments as well. But it does there's a lot of other challenges, right? So if you're in a very topographically heterogeneous environment, you're in the mountains in Colorado, Rocky Mountain National Park or something, looking at limber pine, I mean, you're going to need a drone, and you need to account for variation in angle and all of the other challenges in remote sensing. And so there's still a lot of work that needs to be done to make this usable from that standpoint. But from an operational screening standpoint, where that is what you're doing, you're testing individuals, it works pretty well.
Yeah,
Awesome.
thanks so much. Yeah, thanks so much for the response.
Sorry, thank you. So actually, related to that, Francis asked in the chat, what is the cost of remote sensing setup?
Yeah, that's a good question. So once the hyperrail was set up, there's a group at Oregon State that helped with it, the Opens Lab. And if we remove the cost of the camera itself, So this is a problem, right? Because the head wall camera, the last quote that I had on one, I think it was like $120,000, $115,000, which is really expensive. That's why a camera that just uses the PRI would be so interesting, because it would be much cheaper to do. Now, the hyperrail itself, $300 is about the cost of it. We bought tripods and these wheels you might be able to see at the bottom. You could put some motors on those so that they would drive as well. We had to think about how to have this hyperrail move over the irrigation systems and drive between the boxes at Drina. So we do have some good sized wheels on them, and it works pretty well. But just the extruded aluminum, this is all 3D printed. And we have some regular tripods. It's not all that expensive, $300-400 depending on the size that you want.
So cool. I see Sean has his hand up, so I'm going to enable him to ask a question.
Hi, can you hear me?
I can.
Perfect. Great talk, Jeremy. This is all very promising stuff. I had a follow-up question on your answer to Enzo's question. I agree that it seems wildly impractical to try and take the hyperrail out into actual ecosystems, but I was wondering about satellite data. You mentioned that there was promise that PRI would be able to be used as a predictor, and I believe you can get that from MODIS data. And so I was wondering what your thoughts were there on satellite-based.
Yeah, of course, I think that satellite-based approaches would be good. I think the issue comes down to the area that you're resolving to in terms of the scale of the image, right? So MODIS is going to be whatever it is, 500 meters or something, or half kilometer, right? In terms of the spatial resolution. You're not going to resolve single trees at that point. So yes, you could calculate PRI, but unless an entire stand is infected. So you could identify outbreaks, like big areas that have been impacted, but in terms of individual trees, you'd have a harder time. That's not to say that it's not going to be possible. I know on the International Space Station, they're starting to put on their antennas, they're going to have hyperspectral remote sensing. They're going to have all kinds of sensors that would potentially, if we could also unmix pixels and things like that, it might actually get us closer to doing that. So
great, thank you.
Hopefully in the future.
Yeah.
Yes, awesome. All right, we have another question in the chat. I'm afraid of mispronouncing Moshta. Is that right? Let me know. They say, thank you for your talk. I was wondering if there are any risk of infections to surrounding outdoor areas, pine trees, as you infect and grow the seedlings to look for resistant phenotypes.
Well, so the reason that Dorina is partially why it is where it is because when white pine blister rust was introduced around British Columbia, I mean, it came through the Pacific Northwest and took out the western white pines and the sugar pines and most of the white pines that were susceptible around there. So blister rust itself is endemic there and is everywhere. And so it's not introducing anything that's not already around. So we don't really have any risk of infecting any other white pines in that area. Now, if I were to, if you wanted to bring it to, say, the Southwest, where southwestern white pine is, there's not a lot of infection there yet. We would never do that. We would never bring and infect trees there to test in their natural environment.
Thank you. All right, John has a question, or I think it's more of a statement. So he says we plan to use a drone next week for structure and spectral analysis in the High Roberts fire 2002 burn. White bark pine in the Maller are in the open with wide spacing, generally speaking. So High Roberts is in the Strawberry Mountain wilderness.
Yeah, so suggesting that it would be a good place to test the approach using a drone. Is that what you mean?
I'm not sure. John, do you want me to enable you to speak? Well, thank you. Any other questions for Jeremy? All right, Betsy is asking if you know if cameras that capture PRI are currently available for drones and approximately what that would cost. Very
Yeah.
good question.
Oh, I think that's a good question, Betsy. We've been talking about this. So, and you'll know, Betsy, I mean, John Selker is involved with this project as well, and Chris Still. And when I talked to, I'm trying to remember who gave me the quote. It might have been John, but he said that an individual sensor. So if you're building one of these cameras, you can basically get one of these individual sensors. I'm not sure what the width of the spectrum would be, but it's something like $12 for an individual. And so if you could build this type of thing like we do with Raspberry Pis and Arduinos reasonably cheap. It would have to be calibrated and some stuff. And I'm not exactly sure what a prototype would cost, but it would be fun to find out. I'd have to get a quote from somebody to build one, but I think that it would be reasonably priced. And then, yeah, you could put that on a drone and do all kinds of things with it. I don't have an exact cost. I wish I did, but I know that the sensor is around $12 for a single band.
She says, so you're saying there's a chance.
Yeah.
That's really exciting.
Yeah.
I'm very excited to see where this goes.
Yeah. And again, like we said, that the head wall camera, $100 and some $1,000, it fell out of the sky on a drone and it cost $15,000 just to fix. And so that's sort of scary from the approach that these pieces of equipment are flying around on these drones, and
right, it's
risk.
pretty high risk. Yeah. Diana is asking if Worldview 3 imagery would be useful for this application.
Yeah, it might be. Now you use Worldview, don't you, Laurel?
I do, yeah. My thought is that with multispectral imagery, which is the Worldview 3 imagery has 8 bands, that it probably won't be high enough spectral resolution to catch those subtle differences, but we could try. The other trick would be having large enough individuals that are infected and of known infection, like duration of infection or severity of infection. Yeah. So that data set would take a lot of time to amass.
I think you could find that though.
Yeah, you definitely could find it.
The Lincoln National Forest with Southwestern, all the trees are basically, well, I won't say all of them. A lot of them have been tested. We know what the level of resistance is there, and basically know when infections started in the 70s. I mean, you could probably figure out an area there to test it with. And they're big. Some of the trees are big.
And with the hyperspectral work that you all have done, you've identified features that we could then try to develop indices or band ratios specific to Worldview 3 that could potentially differentiate infected trees from uninfected, but it probably would need to be a more severe infection.
Yeah, it's interesting to think about.
Yeah, definitely. It would be worth pursuing because it's cheaper, potentially, over a long distance. Other questions for Jeremy? All right, thank you so much. That was an amazing talk. Well done.
Well, thanks for inviting me. I really appreciate the chance to visit with everybody and kick off this seminar series. So
yeah, thank you. All right, the seminar commences the 3rd Tuesday of every month, so be sure to come back and see our next speaker, who is Tommy McLaren, who will be talking about giving a talk on understanding Clark's nutcracker seed resource use in Yellowstone National Park. So that's October 18th. Thanks, everyone.